So I'm pleased to introduce our first Earl Davie keynote speaker today. Dr. Toni Antalis is a professor of physiology and surgery at the University of Maryland, where she's also the associate director for training and education and the associate director of the Center for Vascular and Inflammatory Diseases. Okay. Toni received her PhD in biochemistry at Rice University, then did a postdoc at Baylor. After a few-year stint in Australia, where I understand she became sort of infected by an interest in serine proteases, she joined the vascular biology research group at the Holland Lab of the American Red Cross in Rockville, Maryland, and since then has been well supported well supported by competitive grants. She's widely known for her expertise and research efforts in understanding how membrane-anchored serine proteases and their signaling pathways influence vascular biology in disease. And I know that Earl would be thrilled that you are giving this keynote.
So thank you, Ed, for inviting me. It's a pleasure to be here, and I'm really looking forward to the rest of the program. I think this is a really very interesting and innovative symposium. So today I want to talk about our work on coagulation and fibrinolysis anchored via GPI-anchored serine proteases. And you may not or may be familiar with the family of membrane-anchored serine proteases, which has been our interest for probably 20 years now. There are 20 human members of this family. Can I use? Yeah. 20 human members of the family. And these proteases are the classical serine protease domains anchored to the cell membrane with various anchors. They can be a C-terminal transmembrane anchor, GPI anchored, or type 2 transmembrane proteases, all with the serine proteases domain sitting on the outside of the cell. They have a diversity in structure, multiple specialized protein modules that are associated with them, and they've have emerged to play critical roles in essential and diverse physiological processes, such as maintenance of epithelial barrier function, regulation of blood pressure, and viral cell entry. And you would probably have heard of the tempress proteins that regulate the spike proteins of COVID-19. So testicin is one of only 2 GPI-anchored membrane-anchored serine proteases that And so we've kind of been interested in what this protease does. So our initial studies on testicin showed that it's highly expressed in developing spermatocytes, very rarely expressed in other tissues that we could detect. And it's critical for sperm maturation, motility, and fertilizing ability when it's knocked out of mice. But we also found that it is expressed in human microvascular endothelial cells that undergo reorganization and tubule-like formation on matrigel and during capillary morphogenesis on fibular-type collagen. Another group actually identified testicin using a microarray analysis and found that it was consistently expressed on large vessels and the microvasculature. But the function of this protease on endothelial cells is not known. So we've been interested in what the role of testicin is in microvascular endothelial cells in angiogenesis. So as you would be aware, angiogenesis is the process of new capillaries growing from pre-existing vessels. It's required for normal tissue growth and repair, and insufficient angiogenesis is associated with a large number of pathological conditions, and including diabetes, stroke, cancer. The vascular integrity of these newly forming vessels relies on the endothelial cells that line the vessels. Now, angiogenesis is tightly regulated and it's quite complex. There are multiple steps and initially that you have an initiation phase, which involves VEGF or other angiogenic factors, inducing an increase in vascular permeability and endothelial cell proliferation. This signaling stops, and you get directional endothelial cell migration and reorganization facilitated by the formation of a provisional fibrin matrix. In the resolution phase of angiogenesis, the endothelial cells coalesce and reform the endothelial barrier. Now, if the angiogenic vasculature doesn't form properly, it's inherently leaky, disorganized, and unresolving. So we wanted to ask, using our testicin knockout mice, does testicin play a role in angiogenic processes? And to do this, we went to the in vivo model of angiogenesis involving vascularization of the corpus luteum that's induced by superovulation. So when ovaries are during superovulation, the corpus luteum is an endocrine structure that's formed after the release of the mature ovum. And this structure, endothelial cells invade this structure, and it is a very fibrin-rich ovulation site and forms a primitive vascular network. It's very rapid angiogenesis in this model, and it's akin to what's seen in a fast-growing tumor. We induce angiogenesis in female mice by IP administration of PMSG and HCG. So what we found when we did this model was that the testicin knockout ovaries have a significant increased number and severity of hemorrhages with no difference in endothelial content or in much more severe. And this indicated, while we didn't see an increase in endothelial content, at least in this model, it suggests that there's a functional defect that's involved here. So to try and delineate this more specifically, we used Evans blue dye, which can invade, for leaky vessels, invade into the tissues. And we did find that ovaries during luteal angiogenesis have increased extravasation of the Evans blue dye that's injected in the vasculature, and also that this is associated with reduced expression of VE cadherin, which is an adhesion molecule essential for the homophilic interactions between the endothelial cells, which is consistent with the increased vascular permeability, suggesting that testicin plays a role in this interendothelial barrier function. In another model of angiogenesis, this is the ex vivo aortic ring assay, we also see impaired capillary growth in ex vivo, and suggesting that, which measures endothelial cell migration, tubule formation, and branching, and we see reduced growth. And when we look at a tumor model, we see aberrant formation of angiogenic vessels. This is a B16F10 melanoma model. They appear tortuous, disorganized, and underdeveloped. So what is happening here? One of the things I wanted to mention was that testicin expression varies in human microvascular endothelial cells, being highest in the brain, which form the tightest vascular barriers. So in vitro, we took an endothelial cell line and we knocked down testicin expression in that cell line and asked a number of questions as far as to delineate testicin functions. Initially, we looked at the permeability of FITC dextran across confluent cell monolayers, and we found that the permeability increased when you knock down testicin, similar to what we saw in vivo. This was associated with VE cadherin cell-cell junctions and expression that is reduced in the microvascular endothelial cells, and looking at protein expression, VE cadherin expression is reduced, and also tyrosine 658 VE cadherin is increased, which is a permeability-inducing phosphorylation of VE cadherin. So we did the classic endothelial cell migration and tubule-like formation on Matrigel, and this is a movie showing you what happens with siRNA control versus the testicin knockdown. And you can see that in the control, you have formation of tubules, whereas in the knockdown, they don't do a whole lot. And if we want to measure tube length as a quantitative measure of angiogenesis, we find that total tube length is quite abolished. We also looked at cell migration using on Matrigel using the assay for directional migration and scratch wound closure. So essentially here you make a scratch with a pipette tip and watch the endothelial cells migrate towards each other. And here you can see, again, while the wild type control form close the wound, testicin knockout is much more deficient in doing that. And again, this is a quantitative assay of multiple experiments. So I want to focus now on fibrinogen, which is a key player in wound healing and tissue repair, and it's important in angiogenesis. So during repair after injury, hemorrhages are prevented by the deposition of polymerized fibrin. Fibrin acts as a signaling scaffold for recruiting endothelial cells that guide the formation of vessel structures that integrate into the surrounding tissues and allow for blood flow to support tissue regeneration. The deposition of fibrin occurs as a result of, as you will all know, cleavage of fibrinogen by thrombin to induce its polymerization and cross-linked fibrin networks. And thrombin is produced by the activation of the prothrombin zymogen by factor 10A, either through the extrinsic or intrinsic coagulation cascades. The persistence of fibrin is regulated by fibrinolysis, where plasmin is key, and plasmin is generated by plasminogen activators. And as healing progresses, fibrin is remodeled, the fibrin matrix is replaced, and you get collagen deposition, and this helps to form the newly formed vessels. So the increased hemorrhages observed in the corpus luteum of the knockout mice during luteal angiogenesis, the impaired directional migration and absence of testicin in vitro not only imply endothelial cell barrier leakiness, but suggested that testicin may alter the deposition of provisional fibrin matrix. And so we asked this question, does testicin intersect with hemostatic or fibrinolytic protease cascades? To do this, we developed a cell-based functional assay to monitor fibrin generation and degradation on the cell surface. We took a cell line that are plated on 96-well trays and single washed, which leaves trace amounts of serum components on the cell membrane. And the adherent cells are provided with fibrinogen and prothrombin zymogen to monitor fibrin generation. And we monitor the conversion of fibrinogen to fibrin by the increase in turbidity at OD 350 and analyzing urea solubilized lysates to look at the products. So once fibrin is formed, we can also monitor fibrinolysis, and this is also detected by the decrease in turbidity and the appearance of fibrin degradation products. So when we overexpress testicin, this is addition of function in a cell line we used here testicin, what I call wild type testicin, which is the GPI anchored form, and a mutant testicin where the catalytic serine is replaced with an alanine. And we did this assay in the presence of prothrombin and fibrinogen. We found that whereas with control cells, you see a normal curve where you get formation of the fibrin polymerization and then fibrinolysis afterwards. With testicin present, both processes were accelerated. You see a faster increase in clot formation and more rapid fibrinolysis, and this is quantitated here. So we first then said, well, what's happening during fibrin polymerization? And we set up the assay so that we monitor that initial phase over 5 hours and with prothrombin and fibrinogen. And we find that, that we do get cleavage into, or we get the cleavage to form for fibrin polymerization, but the testicin mutant doesn't do this. And you can see that here with turbidity and also with the Western blot analysis, showing that catalytically active testicin is required for this process. We also looked at the presence of whether prothrombin was required for testicin fibrin polymerization. And indeed, if we leave out prothrombin, we don't get this effect. We must have prothrombin present, which indicates that that testicin is acting upstream of prothrombin. Does it catalyze? So we wanted to ask then, above thrombin, is testicin acting? And we used factor 10 inhibitor, rivaroxaban, as a highly specific factor 10A inhibitor. And you can see that under our conditions, rivaroxaban is a potent inhibitor of factor 10A, but it doesn't inhibit testicin, and it also doesn't inhibit testicin effects on the cell surface. And when we added rivaroxaban to this cell assay, we found that indeed it does inhibit the formation of turbidity and also the polymerization of fibrin, which says that testicin is acting upstream of factor 10A. Does testicin convert factor 10 to factor 10A directly? And the answer is no, that we couldn't find cleavage of factor 10 to 10A or activity in the presence of testicin. So this leads us to conclude that testicin is acting upstream of factor 10A, and we are currently looking at how this might be happening in terms of a very complex coagulation cascade. Now, does the other side of that coin was the fibrinolysis? Does testicin accelerate fibrin degradation, or how does it do this? And so we find that if we set up the assay so that we get clot formation and then monitor fibrinolysis, that if we add tranexamic acid, which inhibits plasminogen activation, that we abolish the effect of testicin. Again, showing that the function of testicin is upstream of plasminogen activation. Now, or it requires plasminogen activation. So we look then, does testicin activate plasminogen? And the answer is no. It is not a plasminogen activator, so it does not cleave plasminogen directly. And you can compare plasminogen activation with testicin versus UPA. This is using substrate activity. Then we looked at whether testicin might directly activate a plasminogen activator, and we looked at UPA because UPA is the cell surface plasminogen activator as opposed to TPA. And we found that testicin actually directly cleaves pro-UPA zymogen similarly to plasmin, and also produces catalytically active UPA. And this is, the UPA activity is produced on cells as well. And so that this suggests that testicin on the cell surface can modulate plasminogen activation and induce plasminogen-dependent fibrinolysis. So finally, the loss of testicin results in multiple large hemorrhages during luteal angiogenesis and impaired capillary formation. It increases vascular permeability due to the loss of testicin due to the loss of VE cadherin, and it's localized on the cell surface where it is available to actually modulate multiple pathways involving fibrinolysis, coagulation, and barrier function. And so we think that testicin may activate or regulate previously unrecognized zymogen activation pathways. And this is only one of these masks. And so several of the other masks may actually play roles in these processes as well on cell surfaces. So I just want to acknowledge the people who helped me helped with this work, and Marguerite Buzos is the main research associate who has done a majority of the work. And I want to then thank the funding agencies, and thank you for your attention, and happy to take any questions.
Thanks, Toni. Yeah. Thanks, Toni. That was fabulous. I have a question about regulation of testis and these membrane-associated serine proteases. So are there natural serpins? Are they regulated or enhanced in terms of anions, polyphosphate, heparan sulfates? What do we know?
We don't know very much about that at all. In fact, testicin, what activates testicin on the cell surface is not known. And we've looked at a few things, but haven't been able to identify what actually activates. Now, with matriptase and some of the other proteases, activation of them can occur. These are the type 2 transmembrane proteases through phosphorylation of the internal tail. But I don't think that happens with testicin, so we're not sure.
That was really interesting. I'm wondering if the UPA receptor is available in the context of testicin, and if that may accelerate UPA activation in the context of testicin, or whether that may in fact have anything to do with testicin activation.
It's a really interesting question, and we've actually been wondering about that too, because the UPA receptor is a GPI-anchored protein too, and so the two of them may co-localize on lipid rafts, and that may be where this acceleration happens. But yes, these cells contain UPA receptor, and so there is a real possibility that testicin and the UPA receptor have some communication as well as with you, because UPA acts on the cell surface through this receptor.
Thank you.
Lots of questions. I may have missed it, but you said it doesn't activate 10 directly, but does it interact with tissue factor 7A to activate 10?
We're looking at that. That's a really interesting question. It's been a little bit difficult to get confirmation, but yes, we're looking at whether that's the next step up from where we're factor 10, yeah. And tissue factor obviously is going to be important for the cells.
And expression profile?
Sorry?
Sorry. The expression profile of testicin, is it also, I'm curious if it can be expressed or is expressed on adipocytes. Have you ever looked? On fat cells.
Oh.
That's a personal interest. I'll talk to you later about it.
We haven't looked at fat cells.
Anybody else? Well, if not, thanks.
Thank you.